Well, good morning, everyone. It's my pleasure to kick off this morning's session and talk to you about granzyme K, which is a new therapeutic that will block chronic inflammation across a lot of diseases. In particular, I'm going to focus on autoimmune diseases this morning. Well, first, the unmet need in autoimmune diseases is significant because current therapeutics essentially just reduce disease activity. There's no cure, and many patients don't achieve remission. Using RA and Crohn's disease as two examples, rheumatoid arthritis, about half the patients respond to any targeted therapy and get about half better. And then the challenge is they only have about a 15% response rate to any subsequent therapy. Crohn's disease, kind of similar. About a third of patients don't respond to initial targeted therapy. Some of those that do lose that response. And actually, surgical usage and hospitalization has not really been improved in Crohn's disease for stricture and fistula and things like that since the invent of biotherapeutics. So where did granzyme K come from? We're doing what we call disease deconstruction, single-cell RNA sequencing analysis of the involved end organs in chronically inflamed autoimmune diseases. This is a T-cell UMAP from rheumatoid arthritis from 80 synovial biopsies, CD4 T-cells on the left, CD8 T-cells on the right. And when you think about the CD8 T-cells, you think about CTL, cytotoxic T-cells that express granzyme B. That's the UMAP showing the expression of granzyme B, and the circles, the cytotoxic T-cells. They're very important. Granzyme B is delivered into target cells by perforin, cleaves caspases, drives apoptosis. That's how we kill virally infected cells in cancers. Very important. But that was not what most of the CD8 T-cells were in rheumatoid arthritis synovium. That circle shows where most of the effector and memory CD8 T-cells were, and they expressed granzyme K, something we don't know so much about. And it was not subtle, as you can see at the red arrow, just kind of a whiteout. Nearly all of the effector CD8s had granzyme K. And so this raised the question, what does granzyme K do? And what we figured out is that it activates a new pathway of complement, which is very important in driving inflammation. To remind you about complement, in the middle is something called the classical pathway. That's where there's a molecule called C1q that binds a protease, C1s, and delivers it to a cell surface, a microbial surface or a cell surface. And then the C1s is an enzyme that cleaves complement components, C4 and C2, to generate what's called a C3 convertase that can cleave C3, generating bioactive fragments, and that can lead to then C5 convertase generation and the generation of C5 cleavage and the MAC, or the membrane attack complex. Next to it is the lectin pathway, a similar kind of approach, except that initial protease is different. It's MASP2 instead of C1s. In the alternative pathway, it's complement factor D. And what you see over to the right is that granzyme K, without any other molecules, can bind to cell surfaces and then cleave those same complement components, C4 and C2, generating C3 and C5 convertases and activate the entire pathway of complement activation. Here's just one slide of the biochemistry showing this. The schematic shows either C1s from the classical pathway or granzyme K, our molecule, or granzyme A as a control, cleaving C4 and C2 to generate the convertase that can cleave C3 and then create C3a as shown on the right where the arrows are, or C3b as shown on the bottom. Note that granzyme K does this, but not granzyme A. It's highly specific and similar to the activation by the other pathways. So when we put the whole story together, these T cells, CD8 T cells expressing granzyme K, accumulate in very large numbers in these inflamed tissues, as I showed on the first slide, and then they release granzyme K that binds to cells that are actually producing complement in those inflamed tissues. And that local concentration of granzyme K coming out of the T cells and complement being produced right in the inflamed tissue produce a tremendous opportunity for complement cleavage and activation. And then that generates C3a and C5a. These are called anaphylotoxins. They bind to their receptors that activate myeloid cells, and they're potent chemotactic agents that recruit a lot of other inflammatory leukocytes. You also get what's called opsonization. That's shown with C3b bound to endothelial cells. That's important for targeting cells and antigens for phagocytosis uptake. Plays a big role in B-cell activation, antibody generation, antibody presentation. And then the membrane attack complex, or the MAC on the right, can lyse cells or activate them when it's sublytic. And so this process of granzyme K being released from these CD8 T cells in the tissues provides all of these pathways of activation mediated by complement that's very profound. Well, is this important in vivo? So that's shown here, where we carried out a model of psoriasiform dermatitis using imiquimod on the shaved skin of mice. And if you look in the middle picture, you see in granzyme K wild-type mice, you see redness, thickening, scaling, characteristic changes of psoriasis. Right underneath it, in the granzyme K deficient mouse, the skin looks nearly normal. That's shown quantitated on the left. And then on the right, we found that this correlated closely with a significant reduction in complement activation in the tissue. And that's shown for C3d and C4d, which are the active breakdown products of complement. They were significantly reduced in the granzyme K deficient mice. Well, this is important not just for rheumatoid arthritis, but from studies that we've published as well as some from the literature. There's actually a pretty long list of diseases that have an expansion, a significant expansion of granzyme K cells in the tissue. That includes a number of other autoimmune diseases like Crohn's and ulcerative colitis, MDA5 dermatomyositis, Sjogren's, lupus, and scleroderma. It includes neurologic diseases. In MS, granzyme K cells are in the spinal fluid and in the brain, and granzyme K is a significant signal in Alzheimer's disease. Really interestingly, one of the most striking findings of immune aging is the expansion of granzyme K cells, suggesting it may be responsible for inflammation, that inflammation with aging that's part of getting atherosclerosis, cancers, and other things. It's also been defined in allergic diseases, and it's found in multiple cancers. So in summary, we've made a fundamental discovery that this granzyme K in CD8 cells are highly expanded in the inflamed tissues across a lot of diseases, and that granzyme K protease activates the complement pathway, inducing profound inflammation. This makes it a compelling target for therapy because if you blocked it, you'd block the tissue complement activation, but you leave intact the other complement pathways for host defense. We've developed multiple compounds that block granzyme K and have desirable affinity and other characteristics. We have patents filed, including compositions of matter, and we're now seeking seed stage funding to move forward in lead candidate development and indication selection. Thank you.